Hello! Welcome back. Today we're going to be discussing the cell cycle and mitosis again, and you actually hit two times, Mr. Kelly. A lot of the material in this chapter, as you read, was very similar to what we talked about last year, so I'm going to use last year's video and then an old me, and then add some new information about the new material that you need to know. So it's going to go back and forth between a little bit of time traveling between me last year, or whenever that video was recorded, and me this year. One of the questions we get a lot is, why don't cells just continue to grow? So why do cells actually need to divide? Why can't we just have one very, very large cell? And there's two main reasons why cells can't just can't just get bigger and bigger and bigger. One of the issues is that as the cell gets bigger, the demand on the DNA is just too great. So you only have so much DNA in your cell that's going to make proteins and control the activities of your one little cell. So if the cell gets so big that there's so much happening inside that cell, the DNA can't possibly maintain it, and it's going to overload the system and the cell is going to die. The second issue is in terms of the actual ability to move materials in and out of the cell. As the cell gets bigger, the volume of the cell increases much faster than the outside cell membrane surface area. So one of the main issues that big cells have, and why they're forced to divide eventually, is that they physically cannot get enough materials into and out of their cell to maintain that volume, to maintain that size cell. So the bigger the cell gets, the more materials it needs, but there's not enough surface area, there's not enough area on the cell membrane to actually allow those materials to flow in and out. So cells are forced to divide and make smaller cells that are more easily able to maintain themselves and maintain what they need to get in and maintain what they need to get out. This next part is a little bit of vocab. So one thing that you should have heard before is chromosomes. So chromosomes are what's actually passed along in between the cells, which is going to carry the genetic information from one generation to the next. So when you pass along your genetic information, you're passing along your chromosomes. So your chromosomes are made up of DNA. And before each cell can divide, you need to duplicate your chromosome. So you can see in the picture here is what your chromosome looks like, an unduplicated copy of it. So you have all your genes, all your actual traits that make up you. And before you can actually make a new cell, you need to double up on your DNA. You need to duplicate your chromosome, because when it divides, each new cell is going to get 1/2 of the duplicated chromosome. So 1/2 of this identical copies. 1/2 will go to one cell, 1/2 will go to the other cell. Each of these DNA molecules are made up, each of these chromosomes are made up of DNA molecules. So all chromosomes are DNA molecules that are tightly compacted and twisted into these shapes. So DNA is a big long string. As we wrap it and tightly combine it, we can create this chromosome structure and shape. Each individual duplicated chromosome consists of identical sister chromatids. And this will get pretty confusing in class, because we call unduplicated chromosomes chromosomes. So if there's just one of them that's unduplicated, we call it a chromosome. But when you duplicate it and make an exact copy of it, we still call that a chromosome. However, that duplicated chromosome has two exact copies of two sister chromatids. So chromatids are exact copies of each other, and together they form one duplicated chromosome. The area where they're joined, so there's one meeting point where the two of them are stuck together, we call that the centromere. So the two sister chromatids, each individual one that's identical to each other, are held together at the centromere. One thing just to remind you about in terms of chromosome structure is that each cell has a large amount of DNA. And as I did mention, it has to be tightly compacted and wound together. And those proteins that the DNA is wound around are called histones. So when you have your DNA molecule, and when it actually is being stored in your cell, it has to be tightly wrapped around these histone proteins, which then form this chromatin structure, this beads on a string structure, which is the common structure and the common shape that you see in a cell interphase, which is like that ball of string that you see. And then eventually during mitosis, that chromatin state, that ball of string, gets condensed even further to actually create the chromosome structure. So all of this is just DNA that is so tightly wound together, it actually forms a physical shape like this. Within chromosome structure, one thing that we do have to add is the ends of the chromosomes in eukaryotic cells and linear DNA are what are called telomeres. And telomeres have an extremely important role in terms of replication. One thing that we didn't really mention last year is that when you're replicating linear chromosomes, there's an issue at the ends. So one advantage of a circular chromosome is that there is no end. So if you start at one point, you can just wrap all the way around and complete the entire circle. Well, in linear DNA, since it's lines, there is an end point. And at the end, the DNA enzyme, the DNA polymerase that makes the DNA, isn't so great at making those ends. It's actually impossible to make those ends, which means that every time you're replicating your DNA, you're actually losing a little bit of that DNA. And those telomeres is that part of that DNA that you're actually losing. So in terms of evolutionary biology, our cells got smart in that they basically put these repeating units at the end of our linear chromosomes that every time the cell replicates, it's chewing away, but that there is no important information there. It is just DNA there meant to be chewed up so that you're not losing important DNA every time you're creating more cells. So if we look at just a cartoonish model of the linear chromosome, we can see here's one of an unduplicated chromosome. You have the centromere here in the middle. All this good light blue DNA is what you need. And then at each end is what we call the telomere. And it's just repetitive DNA. It's just a whole bunch of repeats. And basically they form like a cap structure meant to physically cap off the end. And this is what is going to be chewed up every time the cell is actually replicating its linear DNA. Once again, this is only a linear DNA problem. If you're a bacteria with circular DNA, you're golden, you're good. You don't have to worry about this issue. So if we actually look at the duplicated chromosomes, these with fluorescent dye, it's these greenish dots that actually represent the telomeres. So they were able to stain them and then use an electron microscope actually visualize them. And as I mentioned, the issue is, it's called the end replication problem, is that when the DNA polymerase binds and starts making the DNA, there's always a little bit at the end that basically think about where the enzyme physically has to sit. It can't start at the very end. It has to physically bind to something. So by physically binding to something, basically its fat butts in the way and then it starts moving. So it's the end where it's physically sitting. It cannot physically create DNA at that part. So every single time you're going to lose that little bit of DNA at each of these 3' ends, and it's going to get chewed up. So every single time you can see that you're losing about 50 to 200 base pairs every single time you're replicating your DNA. Some cells have actually figured out a way to avoid this issue. So they actually have an enzyme called telomerase, which does exactly what it kind of sounds like, is that actually builds up the telomeres and replenishes them. So most cells do not have this active. So this is active in your germ cells, so very, very early embryonic cells. And basically they can rebuild up their telomeres so they don't have that issue of eventually, if DNA replication keeps happening, the telomeres will keep shortening. Eventually you'll start chewing up good DNA, useful DNA. Well, if you just keep adding more telomere length, you don't have that issue anymore. So most somatic cells do not actually express this enzyme. Some stem cells, when we discuss later about stem cells, do express this enzyme, but it's highly controlled. And here's the interesting part, is that almost all cancer cells have this enzyme expressed. So one idea is that one of the consequences of aging, and the reason why there's these physical changes as you age, is that, and why you eventually might possibly die from old age, is that basically your telomeres are just so short that you start chewing up useful DNA and you can't make the proteins that you actually need. So there is some anti-aging research in looking at, well, if I turn on telomerase, can I actually keep cells immortal and almost stop the aging process? And it's really hit and miss at the moment. We're still doing active research. But this clearly is a idea of how cancer cells are immortal, is that they can just keep rebuilding their telomeres so they don't have to worry about eventually chewing up the DNA that they need to survive. Now into a little bit about the cell cycle. So the cell cycle is the series of events that the cell goes through. So the cell cycle basically is the entire life cycle of a cell. So it's the entire growth phase, the active living phase, and then eventually if it gets too large, it's going to go through and actually create 2 new cells, so it's going to go through a cell division. The cell cycle consists of 2 main phases. The first phase is known as interphase, which is the period of growth that occurs between cell divisions. So it's between the phase, so it's between the cell divisions. So this is where most of your cells are. Most of their lifespan are in interphase, where they're growing, they're just doing what they're supposed to be doing. And eventually, if they get too large and they reach that spot, they're going to enter the M phase, which is the actual period of cell division. So during the cell cycle, the cell obviously needs to grow. It's going to then prepare for cell division, and it's actually going to do that cell division. It's going to divide and create 2 identical daughter cells. And then the entire cycle will begin again with each of the 2 identical daughter cells. So the cell cycle has 4 phases. The first one is known as the G1 phase, which stands for the first gap phase. Then you go into the S phase, which we'll go over each of these, what happens in each of them in detail in a second. And then the third phase is known as the G2 phase, or the second gap phase. All 3 of those phases together make up interphase. So interphase has 3 subphases, G1, S, and G2 in that order. After you leave interphase, after you're done with the G2 phase, you then enter the M phase, which is the actual cell division phase. So what actually occurs during each individual phase of the interphase? During G1 phase, so G1 stands for gap 1, but sometimes easier to think of G as in growth. So the G1 phase is the main growth phase of the cell. So it's actually going to increase in size. This is where the cell is going to make new proteins and organelles. So this is really the main growth phase. This is where the cell is going to grow the most after it's a brand new cell. During the S phase, the S stands for synthesis. So the S phase is where you're actually going to duplicate your DNA. So S in for DNA synthesis, so you're creating an exact copy of the DNA molecule. So this is where you go from that unduplicated chromosome to that duplicated chromosome. So S stands for synthesis. DNA synthesis is happening. You are creating more DNA, so you're replicating your chromosomes. And once you enter S phase, you're pretty much committed. So a lot of your cells are going to stay in that G1 phase, but once you decide to, I'm going to make more DNA, I'm going to copy my chromosomes, usually that cell is going to finish the rest of the cycle and actually divide, because there's no point in duplicating your DNA and wasting energy doing that unless you're going to actually make new cells. And then the last phase of interphase is the G2 phase, the second gap phase, which basically this is a preparation phase. So during the G2 phase, the cell is going to prepare the organelles and the molecules required for cell division. So it's going to get everything ready for the cell division party, the M phase party, where it's actually going to do that cell division and create 2 new cells. Once the G2 phase is complete, the cell is ready to start the M phase, which usually stands for mitosis and then cytokinesis, which we'll talk about later. However, in sex cells, that is, the M stands for meiosis, which will go through each of those 2 different M phases in a different section. And then in conclusion, sometimes looking at the cell cycle in this pie chart, in this circle graph, is much easier. So you can see here that your cell you create a new cell, so it enters G1 phase, this main cell growth phase. Then it transitions over into the S phase. At this point, it's committed to creating more cells. It's committed to division. At the S phase, the cell is going to replicate its DNA, create a copy of its DNA. After that, it's going to enter the G2 phase, which is going to prepare for the M phase. And then you actually enter the M phase, and we'll go through each of prophase, metaphase, anaphase, telophase, cytokinesis later. But that is going to be the actual process of creating 2 new cells. As you can see, interphase is clearly the longest phase, so the majority of your cells are interphase right now. And you can see that interphase, once again, has 3 subphases, G1, S, and G2. So the next topic I will talk about is after the G2 phase, what are the actual stages, what do each of these phases actually look like in a cell when the cell is undergoing mitosis and cytokinesis to actually create 2 new cells for your body.